Hi guys, welcome back to my channel. It's Doc Janelle Domangon. The subject at hand is cytogenetics, and this is actually a two-part video. First, we'll be talking about cell anatomy, and then the second part of the video will be talking about chromosomal anatomy. If you're ready, then let us start. Of course, cells are defined as the basic unit of life, and cells basically build up the entire human body. When you put cells together, then they form tissues, which you already studied in the last semester's course of histology. Now, cells are generally grouped into two major divisions. These are your somatic cells and germ cells. So, somatic cells are better known as your body cells. These are the ones that basically become your epithelial cells, cuboidal, columnar, and when they come together, they form the different parts of the body. While the germ cells, these are the cells that are used for reproduction. So basically your sperm cells and egg cells. Now, one of the major differences between these two is the number of genome that they have. So when we talk about germ cells, sperm and egg cells, they only contain one copy of the genome. And when a cell contains one copy of the genome, then they are called haploid. Now, when we say genome, it refers to 23 chromosomes. So the sperm cell contains 23 chromosomes from the father, and the egg cell contains 23 chromosomes from the mother. So after sexual intercourse, if there will be fertilization or the meeting of the sperm and the egg, then the 23 chromosomes of the father will be mixed with the 23 chromosomes of the mother. Thus, from being haploid, it will now become diploid. So all our somatic cells, so all cells of the body, contains two copies of our genome, or it contains 46 chromosomes. Thus, they are called diploid. Now, our cells contains many different organelles. And let me just show all of them to you right now. And we'll be talking about them one by one a little bit later. Now, organelles are basically or comes from the root word organ. So these are minute or microscopic organs that divide the labor in order to sustain the life of the cell by partitioning certain areas or serving specific functions. So each of these organelle, they work differently and they keep related biochemicals and structures close to one another in order for them to interact efficiently. The first organelle that I want to discuss is of course the plasma membrane. So the plasma membrane, as you know from your human anatomy and physiology, is composed of a double layer, or better yet, a bilayer of molecules called phospholipids. So remember these globules that you have here? These circular globules that you have, these are your phosphate heads. And these phosphate heads are described to be hydrophilic or water-loving. So they are called water-loving because they can come in contact with the fluid or the water which is found outside the cell, and they can also come in contact with the fluid that is found inside the cell. Now, in between those two phosphate heads are your fatty acid chains, which are these thread-like extensions here found in the middle, which is sandwiched by your two phosphate heads. Now, these fatty acid chains are called hydrophobic because they are water hating. Of course, when you think about it, you cannot mix oil and water together. So oil is a kind of a fat, so it is hydrophobic or water hating. So the plasma membrane or the cell membrane or the cell wall, all three terms refer to the same thing. Cell membrane, plasma membrane, cell wall, pare-pareho lang sila. Ang ultimate job nila is to give shape to the cell, and it works as a barrier to regulate the passage of ions and molecules in and out of the cell. Of course, there are other structures found in our plasma membrane, such as your proteins, which are the purple structures that you see here. Your proteins are embedded in the membrane, and they function as bridge through which water and water-soluble solutes cross. Now, proteins in the cell membrane, there are two types as well. The integral protein, which is this one, which protrudes all throughout the cell. And we also have the peripheral protein, just this one, only found on one side. No? Also, we have carbohydrate molecules. These are the ones in green, which are found also in the external surface of our cells. And what is their job? They are important for cell attachment. So they act as the glue in order for two cells to stick together. They are also important for cell-to-cell communication. So they act like antennas so that each cell can actually communicate with one another. And they are the receptors for hormones, or they act as parang yung pangalan ng street, no? Para silang mga street names yan in order for the hormones to know that these are the cells that needs to be targeted. The next is the fluid inside the cell, which is the cytoplasm. So the cytoplasm is the fluid portion of the cell where most of the organelles float. Aside from water, it is also composed of salts and various other organic molecules. Next, we have the nucleus. The nucleus is probably one of the most prominent organelle, meaning if we have a microscope and we look at the cells, let's say in low power objective, the nucleus will probably be one of those organelles that we can easily identify. The nucleus is also covered by a layer similar to the plasma membrane, and we call it the nuclear envelope. If we examine this closely, you will actually see again your phosphate heads and your fatty acid chains. No? Yeah, double layer din sila. Now, the difference between the nuclear envelope and the plasma membrane, the nuclear envelope contains these holes, and we call this as nuclear pores. And what is the function of these holes? They function to allow movement of biochemicals. Another structure that is found inside the nucleus is your nucleolus, this one in gray. So the nucleolus is important because it produces your ribosomes. And as mentioned in our discussion last week, your ribosomes are important, especially in the central dogma, specifically in what step? In your transcription. Now, other contents of the nucleus are your chromosomes, which we will study later, RNA, especially during transcription, and the fluid portion of the nucleus, which is the nucleoplasm. Now, almost all the cells in the body contains a nucleus, except the red blood cells. So remember that your red blood cells do not have their nucleus when they mature. Another organelle that I want to discuss with you is the powerhouse of the cell, or the mitochondria. So they are called as the powerhouse of the cell basically because they provide energy. And how do they do this? By transforming chemical energy and transforming it into freed energy so that the energy can be used by the cells in its many different functions. Now, when it comes to anatomy rather, or the structure of the mitochondria, it is composed of two membranes. The inner membrane, the one in yellow, we call it the inner membrane. And then the one outside, the one in orange, is the outer membrane. Okay? Now, the outer and the inner membrane are different because the inner membrane forms folds. And these folds are what we call as cristae. Now, because of the infolding of the inner membrane, it also forms spaces, such as this one that I'm pointing to right now. It also contains spaces, and those spaces are what we call as matrix. Also, before we move on, I'd also like for you to notice this. If you look closely, this is actually DNA. So you are right, and it's also labeled here. Mitochondria also has DNA inside of them. So it's not only the nucleus which contains DNA, but also the mitochondria. Next is our endoplasmic reticulum. So if you will look at the picture itself, we can see that the endoplasmic reticulum is attached or right next to the nuclear envelope. So this is your nuclear envelope, and then you have your nuclear pores, which allows the movement of biochemicals, and attached to it is your endoplasmic reticulum. Now we have two types of endoplasmic reticulum, the one which is made up of tubules, the smooth endoplasmic reticulum, and the one which is made up of sacs, which is the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum, the reason why it's called rough is because they are ribosomes attached to it. And what produces your ribosomes again? Your nucleolus. So that nucleolus found inside the nucleus will release those ribosomes outside through the nuclear pore, and they will attach themselves to the rough endoplasmic reticulum, giving it the rough appearance. The RER is important for protein synthesis, while your smooth endoplasmic reticulum is important for lipid synthesis and also for detoxification. Now, I would like to emphasize this because if we think about it, our liver is important for detoxification and also for lipid synthesis. Thus, it means that our liver contains a lot of endoplasmic reticulum. So, afterwards, since we mentioned last time that protein synthesis happens here in the rough endoplasmic reticulum, once proteins are produced in the rough endoplasmic reticulum, they will be packaged inside sac-like organelles which are called vesicles. These vesicles will then be brought to the Golgi apparatus. So let's look at the picture first before we discuss the function. Here are the vesicles which I mentioned, which comes from your rough endoplasmic reticulum. They will now go to the Golgi apparatus. The Golgi apparatus is again, just like your rough endoplasmic reticulum, composed of membrane-enclosed sacs. And basically, the job of the GA or the Golgi apparatus is for packaging, concentration, and final chemical modification of whatever product is brought to it. So in this case that we have been talking about, if protein is brought to the Golgi apparatus, then whatever minor problems, whatever modification needs to be done to the protein, the Golgi apparatus will fix it and will package them accordingly. Now, after it packages it, it will be released again in sac-like organelles called secretory vesicles. And the secretory vesicles will then go to the plasma membrane so that the protein that was produced inside the cell will be released to the outside. Okay? Now, one of those organelles or one of those vesicles rather that comes from the Golgi apparatus is your lysosome. So by context, this is the Golgi apparatus in an image, and those things that bud off, those things that float afterwards are your secretory vesicles. And if a vesicle contains digestive or hydrolytic enzyme, then that is what we call a lysosome. So basically what it does, the cell gets different kinds of toxic materials from outside, and the lysosome will bind to those toxic material and digest it. No? And as we can see here, extracellular debris were taken inside the cell. It was combined together with your lysosome, and it was basically digested. So remember, peroxisomes are different from your lysosomes. Peroxisomes contain naman your hydrogen peroxide. Okay? Lastly, we have your architectural organelles or centrioles. They are composed of 9 microtubular triplets, which comes in pairs. So if we only have one, then that is what we call a centriole. But if we have two, then that is what we call centrosomes. Alrighty. Now, what does it mean when it says 9 microtubular triplets? So let's count this. 1, 2, 3. So we have 3 tubular structures here. 1, 2, 3, 1, 2, 3, and it so on. So if we actually count those triplets, we should be able to count up to 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 tubular triplets. So each one of them is called a centriole. If we have 2, then we have centrosomes. Now, what are they for? They serve as a cell skeletal system, or basically they give the overall support to the cell. And they are also important, especially when we talk about the cell cycle or cell division. Yan. We'll be able to again go back to our centrioles. So the centrioles are the ones that go to the opposite side of the nucleus, and they are the ones that basically produce the mitotic spindles, which attach the chromosomes and pull the chromosomes towards the opposite poles. No, but more of that once we go into cell division. Lastly, we have the cytoskeleton. And this is a meshwork of proteins and rods providing the overall 3D shape of the cell. There are three types of cytoskeleton: your microtubules, your microfilaments, and intermediate filaments. So this tube-like structures that you see here in blue, these are your microtubules. They are formed by a protein called tubulin. Alrighty? And they are important for the cell architecture and for the movement of organelles. Secondly, we have these things at the bottom, the ones in yellow. These are your microfilaments. And your microfilaments are made up of a protein called actin. And if you remember actin from your musculoskeletal system, they are contractile filaments. And lastly, it's not seen in the picture, we have your intermediate filaments. So intermediate filaments is not composed of only a single protein, it's composed of several proteins, which helps maintain the shape of the nucleus. Let us now proceed to talk about the parts of the chromosome. Now for the second part of our discussion, let's talk about chromosomal anatomy. So chromosomes, as discussed, are highly condensed form of DNA. So ang chromosome gawa siya sa DNA, only it is arranged differently. Because as mentioned, diba, remember your DNA is a very, very long strand. And if it will not be arranged properly, it will not fit inside the nucleus. And so the DNA, what it will do is it will wrap around itself around proteins which are called histones. So in this image, can you look at my pointer? The DNA will loop and wrap around itself around these proteins. And once it does, then they become, or they are called, nucleosomes. So these nucleosomes will further loop around and arrange themselves so very, very condensedly until it becomes your chromosomes. No? So it will become into your chromatin fiber until eventually it reaches the more familiar shape that we know it to be. Usually, we see it to be like this, yung parang letter H almost, no? It appears like that, especially during metaphase. So if we want to be able to see our chromosomes in this form, then we should be able to catch cells during metaphase stage. And as mentioned, humans, we contain 22 somatic, or in genetics, we better call it autosomal pairs. So 22 pairs, or in total, 44 chromosomes plus one pair of sex chromosome. So if it is XX, then that is a female. If it is an XY, that is a male. So in total, 44 somatic or autosomal plus 2 sex chromosomes, total of 46 chromosomes. Now, as mentioned during metaphase, this is how we will see our chromosomes. Now, let's dissect the different parts of our chromosome. First, we have the heterochromatin, which is the darker staining part of our chromosome. So I decided to show everything so I can point it out to you. Ayan. So the darker ones, we call it heterochromatin. Why do they appear darker? Or why do they stain darker? Mainly because there are more chromatin fiber found in these areas. However, kahit maraming chromatin fiber doon o mas maraming ikot ng DNA doon, it is gene poor. Meaning, walang masyadong traits that are encoded doon sa darker staining areas. And why are they gene poor? Because they contain a lot of adenine and thymine. So adenine and thymine pala, they do not carry gene very, very well. So, dito nangyayari sa heterochromatin what is called gene silencing, or yung tinatawag natin na hindi na nagkikarry ng traits yung areas. Okay, their expression is reduced. While the lighter staining parts are what we call as euchromatin. So si euchromatin naman, it is less condensed because it contains more protein coding sequences. Meaning, there are more traits found or encoded in your euchromatin. And why are they gene-rich? Because there are more guanine and cytosine found in those areas. So, it is in the euchromatin where we find yung traits for hair color, eye color, tone of voice, height, shape of nose, shape of eyes, etc. So if there will be a problem in the euchromatin, then it will definitely affect the expression of the trait. Pero kung heterochromatin yung masisira, it will not lead to a very drastic change in trait expression. Another part of our chromosome are your telomeres, which are found at the tips. So yung dulo, no? The upper tip and the lower tip. Those are your telomeres. Now, your telomeres are composed only of 6 DNA bases. And these are TTA, GGG, or thymine, thymine, adenine, guanine, guanine, guanine. Kindly remember those 6 specific DNA bases. Now, as mentioned, since their telomeres are dark staining, they do not carry genes, no? They are gene poor. So what is their function? They act as a biologic clock, which means that your telomeres shorten after each mitosis. So, every time magsa-cell division, napuputol yung telomere natin. So, it will keep getting cut at hindi na siya magre-regrow. And if it will reach its maximum, naputol na siya to a maximum, then magsa-stop na rin yung cell division or mitosis natin. Okay, so yun pala yung nagsasabi, no, kung tapos na yung cell division or kung hindi na magdi-divide yung cell. Pag naputol na yung telomere. So, ang example niyan, like in the bones, di ba? So, after we reach a certain height, minsan hindi tayo lumalagpas sa height na yun kasi hindi na nagdi-divide yung cells natin sa bones. So, bakit hindi na siya nagdi-divide? Because naputol na or kompleto na yung pagkakaputol ng telomeres natin. Okay? The middle part naman is your centromere. So, the centromere is familiar because naririnig natin ito when we study cell division. So, this is the middle region where the spindle fibers attach during mitosis. Specifically, the kinetochore. And later, we'll see a picture of the kinetochore. So, it is highly condensed, as mentioned, di ba? Since it is darker staining, it is also heterochromatin. And it contains repeats of 171 DNA bases. So, aside from understanding our heterochromatin, our euchromatin, telomere, and centromere, we also have other parts of the chromosome. These are the P-arm, which is also the short arm. So, why is it called short arm? Because P stands for petite. So, the next one, so this is a good image, no? The P-arm is the one on top, shorter than the one at the bottom. And then, they are separated by the centromere. Yan, this is a good diagram. So, the Q-arm naman is the longer arm of the two. And why is it called Q? Because Q is just next to the letter P. Para hindi daw makalimutan. So, P is the short arm, Q is the long arm. And you will notice, nakalagay dito sa right, no, may nakalagay band 3 and then at the bottom you also have band 5. So there are band numberings so that we know kung nasaan yung iba't ibang mga traits natin. So, ang band numbering, nagsastart siya from the centromere moving outward. Okay? So, alternating din sila, no? 1, 2, 3, 4. Parang ganun yung nangyayari. So here is a better image showing you the P arm, the centromere, the Q arm, and the band numbering. Kaya natin siya nina-number para malaman natin kung nasaan yung trait. Like for example, 11.23. Ito, no? 11.23. Anong trait yung nandyan? Or anong trait yung ine-express niyan? So that is how our chromosomes are being studied. So if let's say may damage naman sa band 22 ng Q arm, we want to discover what will be its effect on the human body. So this is to summarize everything. We have the telomeres on top and at the bottom. We also have the P arm or the petite arm, short arm, the Q arm or long arm, the centromere which is found in the middle, and we also have the kinetochore, the ones on the sides, the one in green, no? At dyan nag-a-attach yung ating mga spindle fibers. Diba? Remember your sister chromatid? Yan. Naalala nyo pa ba to from metaphase? Maghihiwalay yan dalawang yan. Once they are pulled by your spindle fibers. So, we are able to identify our different chromosomes, all 23 pairs of them, through a test called karyotyping. So, kapag nilatag lahat ng mga chromosomes natin, ganyan yung itsura niya. At kapag meron tayong ganitong image, we call it a karyotype. So, it is numbered from 1 to 22, and 23 is the X and Y chromosome. So in other karyotype images, they even stain them differently so that makikita natin yung bands, no? So kita nyo dito, iba't ibang color yung ginagamit nila. Also, chromosomes can be classified according to the position of the centromere. So there are 4 types. These are your telocentric, acrocentric, submetacentric, and metacentric. We'll talk about them one by one. So telocentric has no P arm, as you can see here. There is no short arm on top. The very end is already the centromere. And I want you to remember that there are no telocentric chromosomes present in humans. Also, we have the acrocentric, which has a very small P arm, as you can see here. Very small P arm, the centromere, and then you have your long arm. We also have the submetacentric, which has a short P arm and a long Q arm. While the metacentric, almost equal yung length ng P and Q arm natin. Sometimes, mas longer pa daw yung P arm, no? Sometimes. So the centromere is exactly in the middle of the chromosome. Sorry, disregard this last bullet here. So here is an image of true-to-life chromosomes. So here, it's right in the middle, almost equal yung length ng P and Q arm natin. So that is metacentric. Here we have a short P arm, longer Q arm, submetacentric. And here we have a very, very small P arm and a normal Q arm, that is your acrocentric. Now, if you will notice, itong ating acrocentric, almost hindi mo makita, no? Parang may naputol na part. Actually, hindi siya naputol. That is what we call a satellite. Yan. So basically, nagkaroon ng constriction, at yung constriction na yon hindi nagsustain. Kaya parang hindi nakikita. So parang sa image, putol siya. Yan. So satellites are blob-like ends at the very end of the Q arm, no? And they form because of a stalk-like formation brought about by the constriction. So they do not stain, thus they are not seen in the karyotype. And what is unique about your satellites is they contain many copies of genes encoding for ribosomes. So, itong mga maliliit na to, sila pala ang nag-uutos to produce ribosomes. And they are found, yung satellites natin, they are found in chromosomes 13, 14, 15, 21 and 22. And just to prove a point, here is an image of chromosome 21 and 22, showing you their satellites. And in this image, we have 3 copies of chromosome 21, which is abnormal. There should only be 2 copies. So this is your Down syndrome or trisomy 21. But more of the diseases later on in the semester. So just a few things to remember. First, homologous chromosomes are not identical. So here, we have an image of homologous chromosomes. So ito, 'di ba? 2 copies. Sabi natin 1 pair each chromosome. So itong pair na to, they are not identical. They can have different alleles of genes, and ibig sabihin ibang trait completely yung dala-dala kahit na pareho silang number 19. They do not necessarily code for the same traits or genes. However, sister chromatids are identical, meaning itong isang kopya na to, kapag hinati natin yan sa gitna, yung left, kung anong laman niya, yun din yung laman ng nasa right. So kapag sister chromatids, identical. Pero pag homologous, meaning pareho lang silang number 19, they are not necessarily identical. So again, your sister chromatids are formed only during metaphase or during cell division rather. So they are attached to each other by the centromere and they remain attached until anaphase. Sa anaphase, di ba, hihilahin na sila towards opposite poles by your spindle fibers. At sino yung humihila sa kanila? Yung ating centrioles. So, once separated, kapag 1 copy na lang, both then, tawag ulit sa kanya chromosomes, hindi sister chromatids. So, how do we get karyotypes? Yan. How do we catch them? Or how do we create a karyotype? First, we get 5 ml of venous blood. Then we add a chemical called phytohemagglutinin. So ano yung phytohemagglutinin? Phytohemagglutinin will cause the cells in the blood to undergo cell division. So it will be cultured or iiwan siya for 3 days. Afterwards, lalagyan siya ng colchicine. Para sa naman yung colchicine? To stop cell division. So after 3 days, those cells are already in metaphase. So we will stop the process para hindi na siya mag-anaphase. We'll put it on a slide, put some stains, and then look at it under the microscope. So under the microscope, parang ito nasa circle lang yung nakikita natin, no? And then usually what scientists do, they print out a copy of the image and they cut out or pinaprepare nila yan. So a karyotype or this image is used to study chromosomes and to identify chromosomal abnormalities. So dyan natin nalalaman kung sobra ba yung chromosome 21, may kulang ba, may naputol ba, etc. So as mentioned, this is another image of the process. We get blood, we incubate it for 2 to 3 days. We add a chemical called phytohemagglutinin to stimulate mitosis. After 3 days, we add another chemical or colchicine to stop mitosis in metaphase. We put it on a slide, we look at it under the microscope, and then we photograph the chromosomes. So we take a picture. Afterwards, we cut out the chromosome pictures and arrange them into a karyotype. So imagine, it's very tedious. You have to take a picture and then you have to make sure tama yung pag-prepare natin. So this is a sample of a photograph karyotype. No? Imagine mo, kailangan mapag-partner mo sila kahit na bali-baliko yung iba, etc. And then again, each of them are not identical. So, challenging na mismo yung pag-prepare sa kanila. Alright? So, here we can see homologous chromosomes, 2 copies for each for a total of 46 chromosomes. But nowadays, ako na-experience ko to when I was in St. Luke's, nowadays, computers do the karyotyping for us. So we just have to take a photo of the chromosomes and the computer will be the one to arrange them in such a way. Ayan. So, it helps if you stain them kasi pag same color, alam mo na same chromosome yun. Yeah. So, in this karyotype, we have complete set of chromosomes. And what is the gender? XX or female. Okay? So, actually, I will post an assignment on your TLC and I will make you make a karyotype. So, I'll provide the different chromosomes. Your job is to cut out and then paste them on a separate bond paper, then make sure that they are partnered well. All right? So that's it. Thank you very much, guys, and God bless.